Hello everyone, myself K. Sanjay, first MSc Applied Microbiology, Sacred College, Tirupattur. We are going to see about Salmonella species in this video. Synopsis: General characteristics, pathogenic species, pathogenicity, virulence factor, pathogenesis, clinical disease, laboratory diagnosis, colony morphology and culture medium, biochemical test, serology, treatment and prevention. General characteristics: gram staining, gram negative bacteria, shape, rod shape, bacilli, motility, it's a motile bacteria, capsule is absent, endospores is absent, respiration aerobic or facultative anaerobic, optimum temperature 34 degrees Celsius to 37 degrees Celsius, optimum pH 6.5 to 7.5, habitat intestinal tract of humans and other animals. The genus Salmonella was named after Daniel Elmer Salmon, a pathologist who ran the United States Department of Agriculture microorganisms research program in the 80s. Together with Theobald Smith, he found Salmonella in hogs that to the disease known as hog cholera. Pathogenic species of Salmonella species: Salmonella typhi, Salmonella paratyphi, Salmonella choleracis, Salmonella typhimurium, Salmonella enteritidis. Pathogenicity of Salmonella species: disease transmission, injection of contaminated food and water. Incubation period is 8 to 14 days. Virulence factors of Salmonella species: endotoxin, V antigen that increase virulence, lipopolysaccharides, invasion protein, coat polysaccharides, somatic O antigen, flagella H antigen. Pathogenesis of Salmonella species: after injection and passage through the stomach, Salmonella species attach to the mucosa of the small intestine. Bacteria remain in endocytic vacuoles and replicate in our body. Bacteria transported across the cytoplasm and released into the blood in our body. Attachment, engulfment and replication is controlled primarily by two large cluster of genes, pathogenicity island 1 and 2, on the bacterial chromosomes. Clinical disease caused by Salmonella species. First, gastroenteritis. Gastroenteritis is the most common form. Symptoms generally appear 6 to 48 hours after the consumption of contaminated food or water. Fever, abnormal cramps, myalgia, and headache are also common. Chronic involvement can be demonstrated. Symptoms can persist from 2 to 7 days before spontaneous resolution. And the second one, bacteremia. All Salmonella species can cause bacteremia. Its risk for Salmonella bacteremia is higher in pediatric and immunocompromised patients. The gram-negative bacteremia, however, localized infection. Localized infection means it affects particular part in our body. Example: osteomyelitis, endocarditis, and arthritis can occur in as many as 10% of patients. The third one, enteric fever or typhoid fever, a febrile illness called typhoid fever. A milder form of this disease, referred to as paratyphoid fever, is produced by Salmonella paratype A. In other Salmonella serotypes can rarely produce a similar syndrome. Fever that starts low and increases daily, possibly reaching as high as 104.9 Fahrenheit, that means 40.5 degrees Celsius. It is pyrexemia. And the fourth one, asymptomatic colonization. The strains of Salmonella responsible for causing typhoid and paratyphoid fevers are maintained by human colonization. And then chronic colonization, the chronic colonization with other species of Salmonella. Laboratory diagnosis of Salmonella species. Microscopic examination. First, gram staining. In gram staining, observe pink colored gram negative rods. And second, motility test. It is motile in nature. Colonies morphology and culture medium. First, nutrient agar. In nutrient agar, colonies are circular, low convex, smooth, grayish white and translucent. Second, blood agar. In blood agar, gamma hemolysis are produced. Gamma hemolysis means no hemolysis. Mechanchi agar, non-lactose fermenting colonies are produced. Wilson Blair bismuth sulfide agar. It shows jet black opaque colonies with metallic sheen. And xylose lysine deoxycholate agar. It shows red translucent opaque colonies with black center. And deoxycholate citrate agar. It shows pale colonies. Salmonella shigella colonies. It shows black colonies. Hectoin enteric agar. It shows transparent green colonies are produced in the culture medium. Biochemical test. Catalase test is positive, oxidase test is negative, urease test is negative, indole test is positive, methyl red test is positive, VP test is negative, citrate utilization test is positive, hydrogen sulfide test is positive, orethine decarboxylase test is positive, triple sugar ion test. In this test, alkaline slant acid but gas produced and hydrogen sulfide produced in the test. Serological test is Vidal test. The abbreviation of Vidal test is widely investigated disease assay laboratory. Treatment and preventive measures. Antibiotic therapy. Infections with Salmonella typhi and Salmonella paratyphi or disseminated infections with other organisms should be treated with an effective antibiotics like fluoroquinolones, example ciprofloxacin, and the second one chloramphenicol, and the third one trimethoprim sulfomethoxazole, and the fourth one broad spectrum cephalosporin. Vaccination and preventive measures. Carrier of Salmonella typhi and Salmonella paratyphi should be identified and treated. Vaccination against Salmonella typhi can reduce the risk of disease for traveler into endemic areas. Most infections can be controlled by proper preparation of poultry and eggs, completely cooked, and avoidance of contaminations of other foods with uncooked poultry products. Thank you to all for watching this video. Thank you.